Good. So thanks, just bear me to be a little bit longer. I know we skipped a break already, and we're trying not to freeze you to death before none of this is relevant anymore. So just a few more minutes to talk about our translational trials here at Stanford for the early detection of kidney cancer and of response to therapy. So I'm a research scientist here at Stanford, a biologist by training, and I work closely with Dr. Alice Fan in urologic medical oncology, who is a close colleague of Dr. Srinivas, treating kidney cancer patients, including those with VHL syndrome. And so what translational trials mean is we take results and findings from the basic sciences, so from the studies of genes, proteins, in cells and animal models of cancer, and test whether they are relevant to human patients and how they can be utilized to improve patient care. And so these two examples that I'm going to talk about today, about the early detection of kidney cancer and about the early detection of response to therapy, kind of illustrate a much broader effort in translational science in various aspects, not only at Stanford, but all over the US and the world, where people do research on cancer, how we can utilize better findings from basic sciences and improve patient care. So with these two trials, we really address two important and yet unanswered questions in the management of cancer. So when we look at the timeline of development and management of cancer, you can see on the very left, it all starts with a tumor that at some point initiates where something goes wrong in some cells in the kidney. A tumor is initiated that then grows until we can detect and diagnose it by imaging and tissue diagnosis, biopsies, or surgery. And then at some point, the tumor needs either to be surveilled and monitored or be treated, maybe with ablation, with surgery, or with drugs. At which point we also want to know relatively early on during the treatment whether there is a response to the treatment in the tumor itself. And so in kidney cancer early detection, the question is really how to find kidney cancer when it's still curable early on, before it metastasizes, in every single patient. And in the second one, where we talk about treatments, how do we really figure out whether treatment is working as early as possible to really avoid ineffective treatment, because no one should receive ineffective treatment. So this slide is really to illustrate how important early detection is. So you see these four different stages of kidney cancer. This is how all kidney cancers are classified and staged. The first stage is a localized tumor within the kidney of smaller than 7 centimeters, and it goes all the way down to stage 4, which is tumors that have spread to other sites of the body. And then on the right column, you see how many of those patients diagnosed at each of these 4 stages are still alive 5 years after the diagnosis. And you can see, and this is representative of all cancers, this massive drop in survival the later the stages at the time point of diagnosis. In fact, when you get diagnosed at stage 1, these 4%, so it says 96% 5-year survival, the 4% that have died within those 5 years typically don't die of the cancer. It's usually cardiovascular disease or other complications. So early detection is really important to improve patient survival. What is a bit particular about kidney cancer and some other cancers is that this is still a really relevant problem, because still about 30 to 40% of patients, and that is not only VHL patients, but all kidney cancer patients, do have stage 3 or 4 at the time of diagnosis. So we really need to move those up. That's called the stage shift. We really want to detect those cancers early on, so that ideally everyone is diagnosed at stage 1 if they develop cancer, so we can manage and cure it. So here's the challenge. Cancer early detection is a bit like looking for the needle in the haystack. So we really, and you can think about this in 2 ways. You really need to identify the needle being the patient with cancer in a population where most people don't have cancer. So overall, cancer is a relatively rare disease. So at any given time point, only about 1 to 3% in the population do have cancer. So we need to find the patients, but then also we need to find the signal in that patient that tells us this patient actually does have cancer. Currently, there are no screening tests for undiagnosed kidney cancer patients, whether it is sporadic kidney cancer or patients who do have VHL syndrome but don't know it. And for patients with VHL syndrome, there are the surveillance MRIs, which do a good job at catching kidney cancer early. But to be honest, no one really wants to have an MRI. They require specialized equipment, radiologist expertise, and not to forget, they are really expensive. So they are a particular burden on the healthcare system. So this is why in cancer in general, for the longest time, there has been this idea of the liquid biopsy. So alternative methods with less invasive cancer screening and diagnosis with a simple blood draw. So if we could only find a signal of cancer in blood and diagnose and detect it by just a blood draw, then that would get us around the MRIs for VHL patients, but also would probably allow us to develop a screening test so we can screen the entire population to figure out the needles in the haystack, so which are the patients who actually are affected with kidney cancer. There have been a lot of efforts, and they're still ongoing, to develop these blood tests, these cancer screening tests. These often focus on tumor-derived biomolecules, so molecules that the tumor actually kind of spits out and releases in the bloodstream. That can be tumor DNA, so the genes or the genetic material that comes from the tumor. Those can be proteins coming from the tumor and circulating in the blood, or entire cells from the tumor circulating in the blood. Nevertheless, you can imagine that when tumors are small in these early stages, these biomolecules, even though they are very specific to the tumor, it's very difficult, or they don't often lack the sensitivity to detect cancer early on, because when tumors are small, they just don't release a lot of material. So what we have learned in the field over the last couple of years is that these biomolecules derived from the tumor are often good to detect late-stage disease, but they have limited utility in detecting early-stage disease. Now, there are a couple of approaches being researched at Stanford and elsewhere how to develop more sensitive methods to find this needle in the haystack. So you can think of things that kind of drag out the cancer signal from the blood and enrich your sensitivity or improve your sensitivity to really find that needle with methods that are just geared towards finding the needle in the haystack. Or you can think of engineering approaches to enhance the cancer signal and make the needle send out a signal that you can just detect more easily. Nevertheless, we focus a bit more on the response of the immune system to cancer, and has been known for a long time that the immune system kind of is a very good sensor for detecting not only cancer, but reacts to various disease states in the body, including cancer. And so the way we think about this is if you have a haystack without the needle, that haystack itself is actually different when a needle is present. So if the haystack is the immune system changes its state when the needle, being the cancer, is present. And so if we could detect that changed immune state, that might serve as an amplified signal for detecting the cancer itself. So a bit of an indirect way of detecting the cancer, but improving the sensitivity to detect it. So these immune responses often result from interactions between cancer and the immune system itself. And as I said, they often result in changes in the entire immune state in a patient. And such changes in the immune system seem not only to occur in cancer, but even seem to be specific to individual cancer types, meaning that the immune state in kidney cancer is different from a lung cancer, is different from a colorectal cancer. Now, when we talk about the immune system, the most important players are typically the immune cells. So here are some examples of them. Most people typically think of the white blood cells, and there is a lot of research going on in lymphocytes or white blood cells, which are the T cells and B cells that do a lot of the job in fighting diseases and attacking abnormal viruses, bacteria, or even tumor cells in the body. But what we are actually more interested in, for reasons that I will go into in a moment, are the platelets, which are usually our first-line responders when you have a wound. So they are the ones that close the wound, but they're also part of the immune system in modulating the immune state. So we specifically look at the gene products in platelets, which are called RNA. So if you remember your biology, high school biology, of genetic material, that's the DNA, which gets translated into RNA that serves as an intermediate to make proteins, and the proteins are the ones that carry out most of the functions in our cells. So here's a platelet in a schematic form. They actually come from the bone marrow, from a cell type called megakaryocyte that kind of shed platelets into the bloodstream all the time. Platelets do not have any genetic material themselves, so they don't have a cell nucleus with genetic material, but they do receive RNA, these intermediates, from their parent megakaryocytes that kind of package the RNA into the platelet that serve them to produce protein and carry out their functions throughout their lifetime. This RNA is used to make proteins, and then research has found that these disease-specific changes in the RNA repertoire within the platelets are indeed, or have a potential to be specific to different diseases. So this has been shown in cardiovascular disease, in autoimmune disease, and more recently also in cancer. So this seems to be something in these RNA repertoires within platelets that indicates the presence of specific diseases in the body. And as I said, we think of this as improved sensitivity because as part of the immune response to cancer, platelets seem to amplify the disease signal through communication of the platelets with the tumor, with other immune cells, and with each other. So Viola Chen from our lab teamed up with Tommy Metzner from urology to do a little pilot study in patients who have a diagnosis of a kidney tumor, 15 in total, and 2 after surgery, where it turned out that they have stage 1 kidney cancer. And so they collected blood samples from these patients before the surgery, so in the state with cancer, and compared the blood to the state after the surgery without the cancer, looking for differences in these RNA repertoires in the platelets, which would then be a candidate for cancer. And so what they found here, so we're looking at these 2 columns. On the left side, you see the patient without the cancer, on the right side with the cancer. And what we call prediction is how well the RNA repertoires in the platelets distinguish these 2 states in the same patient. And you can see that those are really massively different. So there's a clear difference between the cancer state and the non-cancer state in the same patient, indicating there is potential to have a cancer signal in the platelets specific to kidney cancer. Now, what is really more relevant to today's session here is this question. Does this early stage kidney cancer marker exist in patients with hereditary kidney cancer syndromes like VHL? Because most of those patients were actually sporadic, so meaning they had kidney cancer without having an inherited gene defect, but it just occurred because of a mutation in their kidney cells at some point in their life. And so if you really wanted to expand this to hereditary kidney cancer syndromes like VHL, we would need to test whether this marker is also present in VHL patients. So for that, we teamed up with the International Alliance for Cancer Early Detection and teamed up with our collaborators in the VHL clinics in Manchester and Cambridge in the UK. So this is Emma Woodward and James Whitworth. Whenever you go to the UK, names start sounding like right off the Harry Potter universe. The goal was here to really enroll 60 patients with VHL or other kidney cancer syndromes and isolate platelets, analyze their RNA, and to answer these questions, do VHL patients with kidney tumors have the early stage kidney cancer marker, and VHL patients without kidney tumors don't? And so we are almost done. So we actually enrolled 60 patients. You can see we enrolled 15 patients with kidney tumor, 45 patients without kidney tumor. So it's a bit more unbalanced than what we hoped for, meaning 30-30 would be ideal. So we do continue to enroll a bit more to get this a bit more balanced. The analysis of that cohort is not yet done, so I cannot present you any data, but what I really would want to emphasize here that platelet analysis has the potential to lead to a blood test for early stage sporadic kidney cancer and possibly also for hereditary kidney cancer. And so this is an example how a translational study, so analysis and research on platelets in basic science is being translated into human health and hopefully can improve patient care in the future. So we're still actively enrolling VHL patients for a few more weeks until the end of the year. So if you're willing to participate in this study and are willing to give a blood draw, and I see some familiar faces in the audience, so some of you actually already did. So ideally, we would enroll patients with a kidney tumor, but if you don't have one, lucky you, we still accept you for the study. You need to be off treatment. So patients who are on belzutifan at the moment would not qualify for the simple reason that we would not know whether the tumor is actually alive still and gives us a signal of kidney cancer in the blood. And you need to be willing to come to Stanford Healthcare for the blood draw. So we cannot do it outside. So if you have an appointment coming up for an MRI or to see a doctor here at Stanford Healthcare, then that would be an opportunity to donate this sample. So now what about the second question, how to avoid ineffective treatment? So once the decision is made that the tumors need to be treated and monitoring alone is not enough anymore, there was surgery, was the typical one, but now we also have systemic therapy options with the belzutifan or Velireg. So how do we assess response to treatment? So as Dr. Srinivas pointed out, the typical response evaluation is to do a CT or MRI scan after about 12 weeks, so 2 to 3 months, and literally measure the diameter of the tumors to see whether they have at least stopped growing, ideally shrunk. Now, 2 to 3 months waiting without knowing whether a drug is doing anything good for you is quite a long time. And so ideally, we would want to know earlier whether the therapy is actually effective or not. Because for patients who turn out to derive little benefit from the drug and have to endure all the side effects for 2 to 3 months, there's really a need to know early on whether this is doing anything good for you so that patients who do not derive enough clinical benefit can try other treatment options. So this is the blue box is kind of to illustrate the ideal time window where we would actually want to have the first readout to see whether treatment is working. So you may wonder why we don't actually do the CT or MRI scan earlier. So the reason for that is that tumors take a while to actually shrink. So a lot is going on within the tumor initially, but then it really takes about 2 to 3 months to reliably be able to measure whether the tumors have actually shrunk. So this is why you cannot really do the CT or MRI scans earlier, because even in responding patients, the tumors might not have changed in size. So we need other and better methods to assess early on what's going on within the tumors. And one method that we are investigating here at Stanford is ultrasound as an alternative way to assess what is actually the response within the tumor. So if you compare CT and MRI to ultrasound, I want to illustrate why we actually think that ultrasound can do that. So the good thing about CT and MRI is that it can detect tumor shape and size. It can see all tumors within the body. In some cases, it requires contrast agent. We've discussed it earlier, and some of those agents can be toxic to the kidneys. What it cannot measure are any processes within the tumors. So other than shape and size, it cannot really tell you anything about molecules, if the cells are dying within the tumor, or what the blood flow is within the tumor. Ultrasound can also detect tumor shape and size. A special form of ultrasound called Doppler ultrasound can actually measure blood flow within the tumors. And so that's what we are really after here in this particular study. There's no radiation, and ultrasound typically doesn't require any contrast agent. And in cases where it does, these are typically not toxic to kidneys. The one limitation of ultrasound is that it cannot really see any tumors that are covered by bone or close to air. So for example, in the lungs, those tumors you can typically not see with ultrasound. They're just too enclosed in your rib cage, and the interface with the lungs really disturbs the ultrasound, or lymph nodes in the chest are really a limitation. What ultrasound is very good for is for imaging kidneys, liver, pancreas, adrenal glands, and all superficial tumors. So you can see that there is some potential of ultrasound to at least complement CT and MRI in the way how we assess response to treatments, in particular with the Doppler ultrasound that can actually measure the blood flow within the tumors. And we do know that many tumors, before they actually die and start shrinking, the blood flow is actually reducing in those tumors because they start dying. And so if ultrasound was a way to measure this, then we would have a means to early on detect whether treatment is working or not. So we opened a translational trial here at Stanford that was originally open to patients with kidney cancer receiving tumor blood vessel inhibitors in combination with immunotherapy. So those are typically patients with metastatic kidney cancer. But we recently opened it to a second group of patients with patients receiving any non-immunotherapy, for example, belzutifan. So this is where VHL patients now also qualify for this trial. So the study consists of ultrasound exams to measure tumor size and the blood flow within the tumors. The first scan is done before starting the treatment, so we have a baseline and something to compare the on-treatment measurements to. Then the second scan is after 3 weeks of treatment, and the third scan is after 6 weeks of treatment. So it's those 3 ultrasound exams. And then patients will have the standard CT or MRI scan once before starting the treatment, and the standard one after 2 or 3 months of receiving the treatment. And then the goal or the aim here is to really determine if ultrasound can detect reduction in blood flow at week 3 or week 6. And if this predicts the eventual response, meaning the shrinkage of the tumor in the patients who respond. And if that is true, then ultrasound does have the potential to detect response to treatment early on. I'll give you one example of a patient who already participated in the study so you get a better idea of how that looks like. So this was a 78-year-old male with metastatic kidney cancer to the abdominal wall, peritoneal cavity, so that's the cavity in your belly, and the seminal vesicle. And his disease required him to receive a treatment which was a combination of axitinib, a blood vessel inhibitor, and pembrolizumab, immunotherapy. And eventually he turned out to respond to the treatment. So when we did the ultrasound exams, and I'm showing here the ones before treatment at week 3 and at week 6, I hope that you can appreciate, we can see the size of the tumor. So this is the tumor. And so the tumor already was significantly smaller at week 3 and even a bit smaller at week 6. But what was probably even more dramatic was the reduction in the blood vessel, in the blood flow within the tumors. That's the orange-red one. So you can see all these fine blood vessels through the tumor that feed the tumor and nourish it and give it oxygen. And so already at week 3, there's significant reduction of the blood flow within the tumor. We did this in a few more patients. So I'm showing these different colored lines, illustrate 3 different patients who all turned out to be responders to their treatment. And in all 3 cases, you can see that early on at week 3, the perfusion, which means the quantified overall blood flow within the tumor, has already gone down. It went even down further at week 6. Now, that leads us to the conclusion, even though we still have to image more patients, that ultrasound actually can measure reduction in blood flow, and it appears to predict response to therapy. As I said, we need more patients to validate these results. We also need patients who do not respond to treatment to see whether the opposite is true in that population as well. But as I said, we are still actively enrolling patients for this study, and the second group is now open for patients with VHL syndrome as well. So for another 9 months, if patients who have a confirmed kidney cancer diagnosis, so they need to have had a biopsy or surgery to establish that it is kidney cancer, and they need to have a plan to receive systemic therapy, for example, belzutifan, and they need to be willing to have these 3 ultrasound exams here at Stanford. So if any of you is willing to do this and participate in this study qualifying by these criteria, then please feel free to reach out and we'll be happy to talk about more about this study more. So in summary, giving these 2 examples, what I wanted to conclude with is kind of an outlook. If all this worked, if all this came true, how would management of kidney cancer look in the future? So for detection and diagnosis, the standard evaluation is MRI scans for diagnosed VHL patients, but there's literally no test for undiagnosed patients. So if you were able to find a signal of cancer in blood, and if platelets turned out to be that signal that we can validate after a lot more validation in this, then there might be a new platelet blood test that would allow us to catch kidney cancer systematically in every single patient early on when it's curable. And then in the context of assessing treatment response, we might still need the standard evaluation of CT and MRI to really confirm that patients respond to treatment and confirm the tumor shrinkage. But already having an early look at the tumors and reassure that by an ultrasound exam, something beneficial is happening to the patient would not only be good for the patient to know, but would really also allow us to figure out in which cases this is not happening, in which cases the treatment is likely to fail, and those patients might be better off going on to a different treatment. So with that, I want to acknowledge everyone who has contributed, and you can see it takes a village to do all this research, but really a big thank you, and this is why this is so bold down here, is to all the participating patients, because this research really critically depends on patients willing to participate and being willing to engage in research and contribute by donating blood or doing these ultrasound exams. So a big thank you to everyone who has, and big thank you to everyone who is considering participating in our research or in other research going on at Stanford and elsewhere. I'm open to questions. Thank you.
Thank you, Christian. So questions for Christian?
So is the experience level and expertise of an ultrasound tech relevant to the quality?
So yes, so and in fact, it needs to be a certified and ultrasound tech to do the exam, because ultrasound is still a complex imaging technique that requires qualification to acquire good images that can be interpreted in a meaningful way. So yes, definitely.
As a follow-up on that, is a Doppler ultrasound different from ultrasound?
So it's just a way the ultrasound itself is not different. It's just a way what you measure from the ultrasound that you send into the tissue and that is reflected by the tissue. So you might be familiar with the Doppler effect when an ambulance is driving by and it sounds like high pitched, but then fades out afterwards and the pitch just changes. And so we utilize that same effect when the blood is flowing through the tumor. So the ultrasound waves just catch that flow of blood. And so it's really just the method of detection that's different. The ultrasound itself is not.
Well, your research is really fascinating.
Thank you.
Thank you. But for people who are on belzutifan for kidney cancer, I think I saw on an earlier screen that it was 11 months before results were seen for kidney cancer responding. And would it be useful for someone who is taking belzutifan to have a Doppler ultrasound?
That early on?
I'm sorry?
That early on, you mean after starting treatment, if the response kicks in only younger? So it really depends. And in some patients, we see responses very early on. In some patients, you see responses building up over time. So tumors shrink more and more over time. You have these partial responses initially, complete responses later on. And so those measurements are all based on size measurements. But we do know that things change early on the molecular, on the level of protein, on the level of blood flow within the tumor. So there is a lot of potential if we had methods to access that and interrogate what's going on within the tumor early on that we can't see by just measuring sizes, that we actually detect response to treatment early on.